Okay, thanks all for taking time to come in and listen to this talk. We're going to be talking about combined approaches to basic neuroscience research, and I'm George Portugal. I'm an application scientist at Harvard Bioscience. So it kind of goes without saying, but it's a really important thing to start with, which is the fact that studying the nervous system is an incredibly complex task. There are all sorts of cognitive processes like learning and memory or diseases like Alzheimer's or Parkinson's that involve numerous cellular and molecular substrates, environmental factors, genetic factors, and interactions between the brain and other organs. And so trying to fully understand and characterize these phenomena is really difficult to do. And nowadays, when you're looking at modern publications, in the past, like maybe say 20 or 30 years ago, you could find, still high impact journal publications using one or two techniques to attack a problem. But nowadays, to really make a kind of significant impact with your work, you really need to integrate multiple techniques. And the reason why is that if you have a larger hypothesis and if you're attacking that particular question using a variety of techniques and all of those different techniques give you the same kinds of conclusions, that means you have something really compelling and really strong to say. And then, of course, it's also advantageous if you can validate your ideas, not just from a cellular molecular approach to a whole animal model, but also sometimes things go in the other way where phenomena are discovered in a whole animal model, and then investigators try to understand the cellular and molecular mechanisms that underlie them. Okay, so all that being said, although integration is an incredibly important thing for every neuroscience lab, there are lots of challenges that come into play when trying to do this. So one really clear one is incompatibilities between hardware and software. And I kind of have this image here of an electrophysiology rig to kind of illustrate the point because this thing kind of looks slapped together. And it is, it's something that investigators do all the time where they buy some components from one company, some components from another company, and in principle, it seems like these things should work together. But then when it comes to practice, they discover the hard way that it actually doesn't. And so that's a challenge that old labs and new labs always have to deal with. Another thing is the fact that installation and training of new techniques is a time-consuming process. In the past, when labs were a bit smaller, it was easier to do this because you would have a PI who was adept at numerous techniques, all the techniques in the lab. And so there wasn't this brain drain when a new group of investigators joined the lab. The PI was always there and there was this continuity. But nowadays, with a modern lab, you're constantly bringing in new techniques. The PI might know some of them, but only a small subset. And in reality, they don't really have time to keep up with the latest things anyway. They're spending more of their time writing grants and getting funded and all of that. So installation and training oftentimes becomes an issue, even in a lab that's been doing a technique for a long period of time. And then finally, another challenge that labs often face is with data analysis. So it's one thing to collect data, but it's a whole other thing to understand the implications of it, to deal with the finer issues of parsing out trends in your data and making sense of it. And so all of these different things, lead to challenges in basic neuroscience research. So this is a pain point where we really focus on as a global company. And so for those of you that don't know, Harvard Bioscience is a parent company to a number of different brands, some of which I have listed here on this slide here. But it's actually quite more than that as well. But what we try to do, what we really focus on is trying to integrate and try to reduce these incompatibilities for various labs. And we do this by sharing our expertise, not only in the brain and spinal cord and the CNS, but also we have expertise in the cardiovascular domain, in the respiratory domain, and metabolism. And so we really try our best to help researchers bridge all of these gaps in these multi-system investigations and help them to, integrate all of these various techniques together. Another thing, too, that it's important to note about Harvard Bioscience is that when people often come into contact with our company, they may know us from one particular brand, like, say, Multi-Channel Systems, and that's a company that focuses more on cellular and molecular approaches, and as a result, they may think that that's the only thing that really Harvard does, but that's actually not the case. Harvard focuses not only on cellular and molecular techniques, but also whole animal models as well. And so this ability to do validation in both directions is possible. So you can have these cellular and molecular discoveries that are made that you can then validate in whole organ models or whole animal models. And of course, the opposite is also true. So, and at all of these stages, our scientific expertise will allow you to better achieve your objectives. So to illustrate all of this, I'm going to be talking about a variety of different publications that have come out over the last few years from labs that have instruments from us from a variety of different product portfolios, and they've integrated them together to produce some pretty interesting and compelling work. So the first one I'm going to be talking about is the integration of our behavioral portfolio and our DSI telemetry. So on the behavioral side, we have the company known as PanLab, which has a very large selection of apparatus for behavioral research. And then on the telemetry side, we have DSI, which allows you to acquire data from free-moving animals and do stress-free data collection. So this is a paper done by Turley and colleagues, and in this particular study, what the investigators did was they combined two different techniques. They had fear conditioning, and they also had DSI telemetry. So just briefly an overview of what fear conditioning is. It's a very simple classical conditioning procedure where an animal learns to associate a tone and an environment with a foot shock. And after like one or two pairings of that, and what you do then is bring the animal back into the environment where they learn the association, and they'll freeze just in response to that environment. You can also then put them in a completely novel environment, turn on the tone, and you'll find that they also freeze in response to the tone. So in this particular study, they were also looking at other measures of conditioned fear. So they were looking at conditioned cardiovascular responses at the exact same time that they were looking at contextual and cued fear conditioning. So in this image right here, you see on the top, there's the behavioral side, and so that's with the PanLab instruments recording the freezing behavior, and then like panel B over here shows the contextual or the freezing in response to in response to the tone. So as the animals learn, they freeze a lot more in response to the tone. And then on the bottom here, you have data from the DSI telemetry. And so you can see condition mean arterial pressure, and you can see that there are changes in condition mean arterial pressure in response to the tone as well. So it demonstrates that you have multiple measures of condition learning here. And what's really cool about this is that you can integrate both of these instruments together. You can record both of these measures at the exact same time. And this is something that the study didn't particularly do, but it's also possible where behavioral manipulations could be triggered by cardiovascular states or vice versa, because these two instruments are capable of talking to each other directly. They don't have to just operate in a vacuum. So again, that kind of really highlights the fact that our two product portfolios really do talk well together. This is a little bit more work from the same lab. This is Schweitzer et al. And in this particular study, I believe they were looking at extinction. And so extinction is where basically you teach the animals that these conditioned stimuli no longer have any predictive value for freezing. So you just keep presenting the cue over and over and over again, and eventually they stop freezing in response to the tone. And this is also the case for the cardiovascular responses as well. So that's what you can see down here in this lower set of panels. Okay, so another example which I'll be discussing today is combining plethysmography and telemetry. So for those of you that don't know, plethysmography is where you measure various aspects of pulmonary health. You can do this either in a restrained animal model or an unrestrained model. The instruments that we create have the ability to automatically calibrate the system and automatically deliver drugs. And it's suitable for a variety of different experimental settings. So in this particular paper, this was done by Cleary and colleagues in Nature Communications, and they were interested in autism spectrum disorders, and they were looking at disordered breathing, which is a component of Pitt-Hopkins syndrome. That's one of the autism spectrum disorders. And specifically, they think this has to do with haploinsufficiency of a gene called TCF4. And so they developed some conditional mice which have TCF4, and these animals exhibited unstable breathing under baseline conditions, and they also show a blunted ventilatory response to CO2. And so that's what you can see here in these graphs right here. Now, with that exact same cohort of animals, they were also implanting DSI telemetry, and so they were also looking at changes in EMG at the same time. And so you can see that in the mutant mice, you also have abnormal diaphragm and abnormal EMG response. And if you increase CO2, this difference between the groups is magnified. So again, it illustrates the fact that you can take these two measures and do them at the exact same time. This is actually from the same paper as well. And so not only did they look at EMG and whole body pleth, but they also did sleep telemetry as well. So you've got, multiple measures happening at the exact same time. It's also very easy to integrate all of these different measurements under the same platform. And just as before, you could potentially synchronize events from the separate devices. Okay, so in my next example here, what this particular study did was combine in vivo electrophysiology and behavior. And so the instrument that we'll be talking about next is the wireless 2100 recording system for multi-channel systems. This device is somewhat similar to the DSI telemetry. It allows for wireless electrophysiology in free-moving animals. That being said, it has a much higher sampling rate, so it allows investigators to look at single unit activity or field potentials rather than population events with EEG. But that being said, this study could potentially be done with either of these instruments, which is why I have the DSI implant also over here on the slide. So in this particular study by Pinnell and colleagues, they were interested in activity in various brain regions while animals acquired the Morris water maze task. So for those of you that don't know, the Morris water maze is it's an open arena filled with water, and it's usually with milky kind of non-transparent water that you put the animal into. Now, if you put a rat or a mouse into an environment like that, they absolutely hate it. They try to get out as soon as possible, so they just try to swim around and try to get out. Now, unbeknownst to them, there's a transparent platform that they can find somewhere in the arena. So at first, it takes them a while to find it, but then they learn the extra maze cues that are associated with where the platform is. And so even though they can't see it, if you keep throwing them back into the Morris water maze, they will find that platform faster and faster. And so that's what you can see here. And this is the PanLab data here, panels A and B. And so as you can see, it only takes them about 4 sessions, and they've gone from about 30 seconds of time to about 10 seconds of time to find where the platform is. And so again, they're using these extra maze cues to find where to go. Now, as you can see here in the upper left, there is a rat with a encapsulation on the top of its head. That's where the wireless 2100 recording system head stage is located. And so with this device, you can look at single unit activity in a variety of different regions. And so that's actually what you can see in this panel down here. And so they found different areas that were activated in response to acquisition of the task and maintenance of the task. So the integrated benefits of these two technologies together is that you can take complex behavioral experiments like a water maze, which obviously is not very conducive to electronics, and yet nevertheless you can get electrophysiology data in that setting, combining the behavior with our in vivo recording system. You can also do peristimulus time histograms where you can correlate spiking and EEG activity with behavioral events because, again, these two instruments can talk directly to each other. And it's also possible to have dynamic interaction between these two events. Some of our wireless systems can do optogenetic stimulation and electrical stimulation. So you could even have behavioral events triggering stimulation in the animal in real time. Okay, so for my next example here, we're moving from in vivo models to in vitro models. And so we're going to be talking specifically about epilepsy. So this study by Laporte and colleagues was interested in mechanisms of endocytosis in brain synapses. Specifically, it's looking at ALIX, which is involved in endocytosis, and it apparently concentrates at synapses during seizures. So in this particular study, the group started with doing hippocampal slice recordings using ALIX conditional mice. And what they found was with these conditional knockouts, there are fewer seizure events, and there's also faster recovery time when seizure events happen as compared to controls. And so this is patch clamp data collected by our HICA EPC10 amplifier. So with this particular study, the group also did microelectrode array recordings. And so for those of you that don't know, a microelectrode array is a network of 60 or more extracellular electrodes that are placed on glass. So you can put a brain slice on top of that or grow a cell culture on top of that. And the advantage to this technology is that not only do you get a high volume of data, you also get information about network activity, and you can do this kind of recording at the exact same time that you're doing patch clamp. And so they did find significant differences in spontaneous bursting behavior, and they also found differences in 4AP evoked bursting using the MEA approach. And again, that supports the data that they found using the patch clamp recordings. And then finally, in this paper, they actually combined in vivo data as well. So with a different cohort of animals, they implanted DSI wireless telemetry, and they were measuring changes in EEG in the ALEX conditional knockouts. So what they found was fewer seizure events in the conditional knockouts. So again, you have this validation here between the whole animal model and the in vitro model. So the converging evidence from these two techniques really strengthens the publication impact. Okay, so in this particular example, this is a publication looking at early Alzheimer's disease by Califat and colleagues. And early Alzheimer's disease is associated with hippocampal hyperactivity and decreased sleep quality. And this group was specifically looking at a gene called PMCH, which encodes a melanin concentrating hormone, which is produced in sleep-active lateral hypothalamic neurons. And this particular study combined MEA recordings and patch clamp. And so what you can see over here are peri-event roster plots. And so you've got over 100 channels of recording, and you have the three different conditions. You have your baseline recordings, you have the same cells in the presence of MCH, and then MCH in the presence of an inhibitor, and you can see that there are changes across the three conditions here. And again, with the same cohort, you can also do patch clamp physiology. And in this particular example, they found decreases in activity between the control condition and the MCH condition. And so you have complementary data here from the single channel studies, which is done by patch clamp, and the extracellular activity that's captured by the MEA. And as I've kind of mentioned before, these two instruments can be pretty seamlessly integrated together and can even be combined with microscopy. So you could potentially add in something like 2-photon microscopy or confocal imaging at the same time that you're doing these electrophysiology experiments. Okay, so for my next example here, this is a publication by Mitsushima and colleagues, and they were specifically looking at ischemic brain injury. And so what you see over here in this particular example is you have, on the one hand, on the right-hand side, behavioral data. So they would elicit a brain injury and then measure locomotor activity. And then on the right-hand side, you have MEA data looking at activity. And so you can see very clear differences with this sham lesion here. You can see synchronized bursting events, which are indicated by these blue peaks, followed by periods of relative inactivity. In the center column, you see a hypoxic condition where there's markedly reduced activity. There's no longer any kind of synchronized bursting. And then you can see at least a partial restoration of function with RIPK1 here. So again, it's an integration of data from both the behavioral side and also with MCS. All right, so switching gears for a moment, I'm going to be just talking about some new product releases that we have that should be in line with this theme that I'm going through in this conversation about integrating our products with other products and strengthening conclusions as a result. So one that we'll be discussing now is the new Viva Mars mobile activity rack system. So this new instrument is a high throughput solution for measuring drug screening, neurotoxicity, neuropharmacology. You get acute activity assessment in numerous animals all at the same time. It uses higher resolution infrared beam technology. It works primarily with rats and mice. It's well suited for pharmaceutical and CRO environments, but also labs that are generating high volumes of data in the academic space as well. So we anticipate that we'll see more publications using this technology going forward and integrating it with some of the other portfolios that we've discussed earlier in this talk. Okay, so the next thing I'll be talking about as well is recent developments with organoids. And so organoids, for those of you that don't know, are a type of three-dimensional culture that attempts to better model what's going on in an intact brain. So normally cells are cultured in a monolayer, which is just a single static layer of cells. But that being said, that's not the same as the way that neurons behave in a brain, where there's structure and function and three-dimensionality. So more recently, investigators have developed new techniques and scaffolding to create these three-dimensional structures that in principle should behave more like a brain and can perhaps better model disease states. So an example I have of that kind of technology being used with our product portfolio is shown here. So Silvosa and colleagues, what they did in this particular study is they developed an in vitro model of brain injury to look at traumatic brain injury. And so they would grow these organoids in multi-well microelectrode arrays, and they would use this device up here in panel C using magnets to basically replicate a concussion kind of experience with the organoids. And then following these traumatic brain injury events, they could then characterize the changes in firing rate, bursting, and network dynamics as a result of TBI. And the fact that this was able to be done in a multi-well environment allowed for very high throughput data collection and lots of different conditions being able to be examined. We also have other techniques for understanding what's going on in organoids. So in previous studies, including the one I just mentioned, organoids are studied on the MEA, which is a flat piece of glass with electrodes on the surface. And although you can get some information using that kind of technique, there are limitations. One being that if there are cells that are primarily focused in the center of the organoid, you're really not going to get access to them if the electrodes are all peripherally on the glass. It's also the case that once an organoid gets to a certain size, there isn't vasculature, and so the cells on the interior may actually be dead, but there's no way of knowing that. And so what we really need are approaches that allow us to go inside of the organoid and better understand what's going on. And so to that end, we have a new product that has been developed known as the Mesh MEA, which you can see right here. And so it consists of a polyamide mesh with electrodes at each of the intersections, which is what you can see in the upper right-hand panel. So what's really nice about this technology is that it kind of acts as a bit of a cradle. So when the organoid is placed onto the mesh, it rests, but then kind of goes inside of the organoid. And it's also possible to seed an organoid and create one from scratch inside of the mesh. And then that gives you much more of a three-dimensional understanding of what's going on inside of the organoid. So this technology has been recently adapted to the MEA 2100 Mini, but we anticipate further development of this approach. And so another new product that we have recently launched is the SOHO telemetry system. This allows you to socially house animals, and you can collect temperature and activity data continuously without any kind of interference. This also opens up other possibilities for collecting other types of measurements as well. It's a GLP compliant instrument, so it's well suited for pharmaceutical and contract research. And yeah, again, it's something that could integrate with other types of technology that we provide as well. So just to summarize, Harvard Bioscience provides a complete portfolio of products that allows you to meet your research objectives, whatever they may be, whether it's whole animal, whether it's cellular molecular, whether you're a new startup lab, or whether it's a large organization like a pharmaceutical company. Our instruments are not in isolation. They talk to each other, they interact together, and that interaction allows you to get to more interesting, more powerful conclusions with your research. Not only that, we have the technical expertise to back you up so that if it's a new technique, you're not having to start from scratch. You have the support from our team to work with you to get comfortable with acquisition, with analysis, and all the complexities of integrating things. The interactions between our instruments allow for more dynamic and complex types of experiments. And finally, our approaches allow for validation in both directions, both from cellular molecular to whole animal and vice versa.